Thank you, everybody, for joining us today. Today, I'm going to talk about our multi-analytical platforms for phenotyping and characterizing our human biospecimens for downstream IO applications. For those of you unfamiliar with Discovery Life Sciences, Discovery Life Sciences is built on 4 key competencies. The first and the base of these key competencies is biospecimens, both the acquisition, preparation, and preservation of these samples. These samples come in multiple different formats, including fixed samples such as FFPE, viable samples such as PBMCs, bone marrow mononuclear cells, and dissociated tumor cells, which we'll talk about a little bit later in the presentation. We do have full EMR access into all of our clinical sites, so these samples come with a high degree of annotation. We also have a normal donor clinic. Through this normal donor clinic, we have access to biofluids from normal donors, including apheresis products, which is something else we'll discuss today. Off of this base of biospecimens, we have 3 analytical pipelines for which we can help to annotate these downstream specimens. The first is genomics. In this case, we offer a full suite of whole genome, whole exome, and RNA-seq technologies. These also include targeted sequencing as well as long-range sequencing platforms. Additionally, we have IHC capabilities. This is both in terms of the validation and testing of clinical samples at scale on, again, multiple different platforms, as well as in-house pathologists to analyze and interpret that data. Finally, we have a self-services platform. This platform deals with all assays in which a viable cell is the starting material. So this is primarily flow cytometry, although we do also offer cell culture, cell isolations, as well as 10x genomics technologies downstream of these products. For today's webinar, we're going to focus on 3 main learning points. The first point is how we phenotype our apheresis donors and how we use this information for both determining potential donors for downstream cell therapy manufacturing, as well as for different cell isolations. The last 2 learning points will focus on our dissociated tumor cell offering. The second one will focus on how we use flow cytometry to characterize these samples, as well as use them for identifying samples for different downstream platforms. And then finally, the third learning point will focus on our use of single-cell genomic technologies to use for identifying novel biomarkers, again, in dissociated tumor cells. Our apheresis clinic is located directly adjacent to our production facility, which allows seamless transition from the apheresis donor to downstream manufacturing. We currently have 5 chairs in operation, although by the end of the year that will be up to 8 chairs. We're currently using the Spectra Optia as our apheresis machine with ACDA as our anticoagulant. All of our donors are HLA typed by next-gen sequencing for 6-digit high resolution, and all of our donors go through viral screening for Hep B, Hep C, and HIV, as well as CMV and EBV reactivity. Our cell viability is consistently about 95% or greater directly after collection. We do use a do-nucleation dye cell counter, in this case the Nexcelom cellometer for cell counting, and we are ISO 9001 certified. We do offer this apheresis material in several formats, both fresh and frozen leukopaks in either a full, half, or quarter size. We then further isolate these leukopaks down to mononuclear cells, again offered in multiple sizes, or any of the major immune cell populations present in peripheral blood. Additionally, we do culture to generate both macrophages and dendritic cells from monocytes harvested from our apheresis donors. And we will soon be releasing GMB-compliant leukopaks. For more information on that offering, please feel free to contact us at info@dls.com. So our apheresis donors are fully typed at the HLA loci. This slide just gives you an indication of the number of unique alleles we see at each locus to show that we do have a breadth of many, many different alleles that are available. We more commonly ask for HLA-02, 03, and A24. We do have over 100 donors in each of those HLA types, and these can be offered. Usually we have donors that are A02 and then A03 or another allele, although we do have some donors that represent both A02 on both alleles. Additionally, we did validate an in-house flow cytometry assay to also look at A02. reactive donors or AO2 positive donors. This assay allows us to evaluate donors real time and gives us a binary yes/no as to if these donors are AO2 positive or negative, and those are the flow plots shown in the bottom right. Additionally, the donors are classified for reactivity against CMV and EBV. This is done by ELISA. Consistent with other reports, about half of our donor population is reactive to CMV, while the majority are reactive to EBV. This data becomes very useful with the HLA data because we can now classify donors and look for antigen-specific T cells, which is displayed in the right panels here. These are looking at donors that were classified as CMV reactive or non-reactive by serology ELISA. Then using dextramers against a specific peptide from CMV, we can identify CD8 T cells from HLA-AO2 positive donors that were previously classified as CMV reactive or non-reactive. So this allows us to use this data to further hone in on donors that would be suitable for downstream assays that we're trying to run. The real power of what we're doing with our apheresis clinic, though, is our immunophenotyping of all our donors that come through the clinic. So in this schematic shows how that workflow is performed. In this case, the donor leukopak is brought into the lab and translated down into the mononuclear cell fraction. At this point, we use Miltenyi's MaxQuant16 flow cytometer. We have analysis templates that we've set up that the technicians can seamlessly stain the cells and stain for either lymphocyte or monocyte populations within each donor. And therefore, they'll have the ability to know which donors would be good either for shipping fresh or frozen leukopaks that are required to have a specific percentage of each cell population, or for downstream isolation and eventual cryopreservation in-house. So looking at our donor population as box and whisker plots here, you can see there is a lot of variability within the donor pool for each cell type that we're currently profiling. What we've done is use this data to basically set thresholds. And these thresholds, which are displayed in these orange boxes, allow us to define which donors would or would not be good for downstream isolations. And so this allows us to get the most efficiency out of a leukopak and ensures the highest purity of these cells downstream, such that if we do a T cell donor who falls below 50%, we're fairly confident that our efficiency and our purity will be very low, and we try to avoid having any inefficiencies with these packs just to make them the most out of all this material that we do receive into the lab. In addition to immunophenotyping these donors, we also perform functional assays in-house. An example of that is a large T-cell expansion project that we recently completed. In this case, the apheresis donors were brought in, and the entire leukopak was isolated down to CD3 T cells. At this point, a proportion of the pack was frozen down as primary non-expanded cells. A very small proportion of that total population was then expanded using the Miltenyi Transact system over about 14 to 20 days. This gives us an expanded T cell population to provide, which best represents an expanded population used for CAR T cell therapies. Additionally, during this expansion process, we evaluated not only the cell counts and the rough cell size during the expansion, but also the expression of known regulators of T cell function or that are kinetically different throughout the expansion process. And in this case, that's CD25, CD69, and PD1 were the key markers we were looking at. Using this, we can follow the T cell activation throughout the entire study. This allows us to know when is the best time to remove the stimulus to allow the cells rest prior to preservation. It also gives the platform for additional target molecules to be analyzed throughout this entire time course. So in this way, we really use the MaxQuant platform not only to phenotype the initial patient samples to identify which donors are best for which projects, but then also for downstream expansion and functionality tests. For the remainder of the talk, I'm going to focus on our dissociated tumor cell offering. And first, we're going to talk about how we've really hyper-annotated these dissociated tumor cells at a very large scale. So dissociated tumor cells are single-cell suspensions of solid tumor indications. We have also performed dissociations of normal tissue, some autoimmune tissue, and some metastatic tissue. Most of that metastatic tissue is melanoma lymph nodes. This is generated via proprietary mechanical and enzymatic digestion. These cells are then cryopreserved, and this really allows us to have full annotation of the sample prior to using it for any study. While the pathologists are very, very good at identifying what the tumor is upon resection, every once in a while we do find an instance where upon review a colorectal cancer actually turned out to be some sort of non-Hodgkin's lymphoma. And so that obviously is problematic if colorectal cancer is what you're looking for. So we let these cells cryopreserve. The cryopreserve is very good in terms of maintaining viability. After the thaw, it's still 70% or greater. And then that allows for the best annotation and selection of samples for downstream platforms. So what we do for all of these dissociated tissues is after they go through the freeze-thaw cycle, upon thaw, they go through a flow cytometry analysis to classify all of the cellular subsets within the tumor, as well as HLA-O2 status by flow. Again, a binary is the donor HLA-O2 positive or negative. The panel that we're currently running is 12 markers large. We first pregate out all dead cells, and that's one thing that we found to be incredibly crucial with these dissociated tissues, is that the use of a live dead discriminator, be that a DNA dye such as DAPI or PI, or any of the amine-reactive fixable live dead discriminators, is really crucial, as these dissociated tissues have a fair amount of debris that can complicate downstream flow analysis. We also recommend the use of a marker against red cells. We do use CD235A or GLI-A in this manner. For fresh dissociations, there are a lot of red cells present, and these do remain after cryopreservation. The initial flow plots that we look at are plots of CD45 versus EpCAM. This allows us to identify CD45 positive immune cells, which you can see in the bottom right corner of the flow plot, versus EpCAM positive tumor cells in the top left. We do use EpCAM for all of our tumors, with a couple exceptions. Melanoma does not express EpCAM. We validated a couple different markers and found CD146 to be the best marker for identifying melanoma tumor cells. So for melanoma, we do use CD146. For glioblastoma and hepatocellular carcinoma, we have yet not identified a marker that reliably works for us. So when I show some data later, just keep in mind that those two subsets are going to underrepresent the tumor cell fraction, just because we don't have a great marker for identifying that. Following this gating strategy, we further classify all of the immune cell populations by the major lymphoid and myeloid populations that would be present within the tumor. This includes the CD4 and CD8 T cells, as well as the B cell populations. We do look at NK cells. They tend to be the rarest of all the cell types that we are analyzing, so we will gate on CD3 negative cells prior to analyzing those. And then we do look at myeloid cells, both as CD11b positive and then the monocytic or granulocytic cells by the expression of CD14 or CD15, respectively. At this point, we have analyzed over 2,500 unique primary tumor samples. This is a heat map showing the average of each of the cell types that we do characterize across about almost 20 different indications. And we're going to go through this heat map a little bit more closely over the next couple of slides. This does not include any of our normal tissue, our autoimmune tissue, or our metastatic melanoma. So if we first look at the tumor or immune cell populations, you can see that there are two types of tumor cell or tumor populations that exist, those that are highly immune infiltrated and those that are not. We find that lung and renal tend to be very highly immune infiltrated when we have analyzed them, versus something like breast, colorectal, ovarian, which have more of a tumor representation when we've analyzed them by flow. CD4 and CD8 T cells tend to be very standard, and actually the averages come out to be pretty equivalent across all the tumor types, except for prostate cancer, which interestingly enough has a high percentage of CD8 T cells within the immune cell fraction. This was a little surprising to us. However, we went through the literature. There was studies that have confirmed both by flow and by IHC a high proportion of CD8 T cells within prostate cancer. As I mentioned previously, NK cells are very, very rare. When we do see them, it primarily is within kidney, ovarian, or liver cancer. B cells tend to be restricted mostly to gastric, colorectal cancer, although there are some unique patients within other indications that have high percentages of NK cells and B cells. Finally, for the myeloid compartment, mostly myeloid cells are present within brain cancer and ovarian cancer. Brain, this makes some sense with the high component of myeloid cells that's normally present within the brain tissue. Again, this does vary on a patient-by-patient level. Granulocytes are by far the rarest of the myeloid subtypes, and these happen to also be the only subtype that we found to be sensitive to the freeze thaw. As I mentioned, these are averages. So if you look at something like lung cancer here, which upon first look at this heat map would seem to be all immune cells with very little tumor present, this really is an average. So if we look at all of those lung DTCs that we've currently analyzed, you do see that we do see a spread. So while most of the samples are highly immune infiltrated, we do have some samples that do have a high tumor percentage. And then as far as each of the immune percentages are concerned, there are some that are high for T-cell infiltration or myeloid infiltration. And so it really is heterogeneous among the population. So we use this data mostly to help with sample selection for studies that are focused more on T cells or B cells or monocytes. But within an indication, you will have a high degree of variability. After the initial characterization of the DTCs at the cellular subset, we really became interested in looking at what markers specifically T cells express. And this was really driven by the great successes with checkpoint inhibitor therapies like Keytruda and Opdivo. So there are a number of molecules on the surface of T cells that will regulate their activity both in a positive and negative format. So we first focused on addressing whether or not we could identify PD-L1 and PD-1 expression within DTCs to use these to further classify these samples for downstream analysis. To do this, we used the companion diagnostic for Keytruda Opdivo, which is PD-L1 expression by IHC. So in this case, we're looking at 2 lung cancer and 1 kidney cancer patient sample that were stained with PD-L1 by IHC and identified a sample that was classified by our pathologists to be 100% PD-L1 expressing, have no expression of PD-L1, or in the case of the renal, somewhere in between. We then looked at matched DTCs. So these are samples that were generated from the same patients as these FFPE blocks. And when we looked at the tumor cell fractions within these dissociated tumor cells, we found a very high degree of correlation with the FFPE IHC data in that we saw almost complete expression of PD-L1 on the tumor cells for the sample that had 100%, no expression on the sample that had 0%, and a shoulder of expression on the renal sample that when gated on was roughly 20 to 30%. And over all the samples we've done, we see a high degree of correlation between the percentage of PD-L1 positive cells by IHC compared to the percentage of PD-L1 positive cells by flow. The nice thing about flow and the advantage of flow is that it has multiple markers. So we can now get a little more granular as to what other cell types within the tumor microenvironment might express PD-L1. In this case, we looked at the myeloid compartment as defined by CD11b expression or T cells by CD3 expression and found that there was evidence of PD-L1 expression in both of these subsets, although it was very low in T cells. At this point, we've done about 60 samples across the 6 indications that currently receive checkpoint inhibitor therapy in the clinic. And we found that pretty much for every indication, there are some patients that will display PD-L1 positivity on the tumor cell fraction. Across the board, there was some evidence of PD-L1 expression on CD11b cells in all cases, except interestingly enough for renal. And that wasn't due to a loss or a deficiency of myeloid cells in that indication, as that traditionally does have a lot of myeloid cells when we've analyzed it. And finally, CD3 cells had very low expression or very few cells expressing PD-L1, but they were present in all the indications we looked at. Conversely, if we looked at the PD-1 receptor, this case on the immune cell subsets, we saw that most of the CD4 and CD8 T cells within the tumor seemed to have high expression of PD-1. Again, this was across the board in all of the 6 indications that we looked at. It was usually greater than 50% or higher. We did look at the other immune cell subsets within the tumor, including B cells and NK cells. For the most part, these cell types were mostly deficient for PD-L1 expression, or PD-1 expression, excuse me. Although there were some indications, particularly colorectal cancer, where there was evidence of PD-1 expression on B cells and NK cells. And again, this was confirmed by several studies that have been published within the academic literature. So while PD-1 and PD-L1 served as a great case study and a known receptor ligand pair within the tumor microenvironment, we wanted to take a little more unbiased look at some of the other receptor ligand pairs that could be present within the tumor. So to do this, we generated a 14-color flow cytometry panel for evaluating these receptor ligand pairs. So again, this panel first gates out any dead cells by DAPI, any red cells using GLI-A. We then use the same FCAM by CD45 plot to identify tumor and all immune cells. This time when we gate on the immune cells, however, we first gate on the myeloid cells as CD11b positive versus the T cells as CD3 positive. CD3 positive T cells are further defined by CD4 or CD8 into the CD4 or CD8 compartments. The myeloid cells are then characterized by CD14 and HLA-DR, such that the CD14 positive cells will be most of those monocytic cells that are present within the tumor. The CD14 negative cells are generally always those granulocytic cells that also express CD15. HLA-DR was added so that we could identify potential myeloid-derived suppressor cells that might be present within the tumor. And then finally, the CD11b negative, CD3 negative subset was stained for CD19 to identify B cells. This provides a nice internal negative control, as most of these T cell receptors are not expressed on the B cell subset. So it provides us a nice control when we're looking at expression levels. By doing this panel, we have 3 channels left open. We used PE, APC, and the PE size 7 channels as they have the best signals. And that allows us to look a little more in depth at new or potentially novel co-stimulatory and co-inhibitory molecules that might be expressed within the tumor. So again, there are a lot of molecules that can modulate T cell receptor function. To do this by flow would obviously take quite a lot of time and quite a lot of optimization. So to help us further define what cells or what receptors we'd like to look at, we performed bulk RNA-seq on 8 different DTC samples across 4 different indications. So this heat map shows the expression levels of all those co-stimulatory, co-inhibitory ligands and receptors within these 8 tumors. The bottom 4 genes are a T cell signature, which is used to define cold tumors, which are displayed on the left, versus hot tumors, which are displayed on the right. So if we look first at PD-1, PD-L1, PD-L2, we see at the RNA level these are all expressed, which confirms our flow cytometry data. Interestingly, PD-L2 is very lowly expressed, although we did define some expression levels, which was pretty unique. We did look at some of the co-stimulatory molecules. In general, we found the receptors to be expressed, but not the ligands. We did confirm this by flow as well. I'm not going to go through that data today. Instead, I'm going to focus today on the other co-inhibitory molecules that were expressed within the tumor. We saw that there was expression of LAG3, TIGIT, VISTA, and TIM3. And in contrast to the co-stimulatory molecules, the ligands for these co-inhibitory molecules we did find to be expressed, and usually at fairly high levels at the RNA level. So an example of what this data now looks like is we can actually analyze these receptor ligand pairs by flow. We're showing the data here as a t-SNE plot so that we can remove the bias of predating any cell type and looking at its expression pattern. So in this case, we're looking at a lung cancer sample that is very immune infiltrated. The tumor cells are represented in the mauve or brownish color in the top versus the immune cell subsets, which are primarily the T cells, CD4 and CD8, a large B cell compartment, as well as a myeloid compartment. Now, when we look at TIGIT expression, we see that the TIGIT expression is primarily restricted to the T cell compartment, although there is a small subset of cells in the middle that does stay positive for TIGIT. Based on other studies, we believe these to be NK cells. Conversely, the ligand for TIGIT, PVRL2, is restricted almost exclusively to the tumor cells, the myeloid compartment, as well as a compartment of cells that we have largely believed to be fibroblasts and endothelial cells. We did look for PVR expression. We found it to be more lowly expressed than PVRL2 and to be highly patient-dependent. Some patients did express PVR, others did not. So we focused mostly on PVRL2 when we did these studies. Now, all of these studies have focused on primarily using the entire tumor. for flow cytometry analysis, but these single cell suspensions do work very well for any sort of downstream magnetic isolation. We've done both T cells and tumor cells. I'm going to go through the tumor cell data briefly. So we have used Miltenyi's tumor cell enrichment kit. The beauty of this kit is that it doesn't predispose any expression of any molecule on the tumor. Instead, it binds to everything that should not be tumor within the tumor microenvironment, including red cells, immune cells, fibroblasts, and endothelial cells. And basically everything that's not selected would therefore be tumor cells. So in this case, this is showing an example from an ovarian cancer cell sample. Again, looking at CD45 by EpCAM expression, you can see that there's about a 40% representation of both populations. After we put it through this negative selection process, it's greater than 95% EpCAM positive, which is again the marker that we've been using to find tumor cells. Although we don't it doesn't preclude samples that don't express EpCAM from going through this process, and we have done this with melanoma, and it does work quite well. At this point, we've performed this tumor cell isolations on over 100 unique samples on almost 10 different indications. In all cases, we do achieve greater than 90% EpCAM positive cells following the enrichment with a very high viability. And this goes down into something like lung, which is notoriously very immune infiltrated. We still can get greater than 95% viability on these tumor cell enrichments. So this provides us a way to do any sort of magnetic pre-enrichment prior to going into more downstream flow, any sort of cell culture or downstream genomics studies. The final point that we want to talk about today is single-cell genomic characterization of dissociated tissues. So recently we brought in the 10x Genomics platform and have been using that to characterize dissociated tissues at the RNA level. So this workflow is very similar to our flow workflow for characterizing DTCs. The difference here is that we do this on both fresh and frozen samples, and all of our samples go through a QC check prior to being loaded into the 10x Chromium platform. Given the expense and the highly technical nature of single-cell technologies, we want to make sure that all samples are of the best quality and best represent the needs of the downstream projects. So these inclusion-exclusion criteria can include different tumor percentages that are required for the study, different CD4, CD8 T cell ratios, and so everything goes through a QC check on the MaxQuant platform prior to being selected into the study. If it is selected into the study, it'll either be directly loaded into the 10x box or go through either a magnetic or some sort of FACS-based cell sorting prior to loading for different subsets. An example of some of the projects that we have currently running or have completed using dissociated tissues includes a large-scale study on mutation-characterized colorectal cancer samples. These were characterized for KRAS, NRAS, BRAF mutations, as well as MSI status. All of these samples were purified and the entire tumor microenvironment was loaded and analyzed by single-cell RNA-seq. Additionally, we have been focusing on head and neck cancer samples, specifically from the oral cavity, and performing both single-cell RNA-seq as well as TCR and BCR repertoire analysis. We've also been sorting CD3 positive T cells from multiple indications, mostly lung, renal, and colorectal. These T cells have been further profiled for single-cell T cell receptor as well as site-seq. And finally, we've been subdividing CD8 T cells, specifically from renal cell samples, to perform single-cell RNA-seq and T cell receptor analysis. So I'm going to go through some data generated from the colorectal cancer samples, as well as some of the T cell receptor sample data, as an example of what this data looks like. So again, using t-SNE plots, this is an example of a single colorectal cancer DTC sample. In this case, we annotated each population based on the expression of transcripts for each cell type. So the top populations are our T cell populations, either CD4 or CD8. As this is RNA, we were able to identify the Treg population by FOXP3 expression. We were further able to identify two B cell populations. These were identified by the expression of CD79A, but interestingly, one of them did have markers consistent with plasma cells. So we identified both a plasma cell and non-plasma cell B cell fraction. The tumor cells are represented by the brown fraction in the middle, and then the myeloid cells are the smaller fraction represented by the pink. Now, while these t-SNE plots give you the nice cell subsets within the tumor, because this is single-cell RNA data, we now have roughly 1,000 different transcripts that we can look at and see where those transcripts fall within each of these annotated populations. So one example is a lytic enzyme, such as granzyme B. As expected, this is primarily restricted to the CD8 compartment. We've looked at all the other granzymes as well as perforin, and again, these are restricted almost entirely to the CD8 T cells. Nice proof of concept that these single-cell RNA-seq dataset is performing as we would expect. So if we again look at those co-stimulatory, co-inhibitory molecules, particularly PD-1 and the TIGIT family members, we can see that this does correlate great with the flow cytometry data that we previously generated. So PD-1 is primarily expressed by the T cell compartment, CD8 T cells as well as the Tregs, although the CD4 T cells in this specific CRC sample have less expression of PD-1. It is not expressed by the B cells, the myeloid cells, or the tumor cell fraction. So conversely, when we look at PD-L1 expression within this colorectal cancer tumor, we can see that it's largely restricted to the myeloid compartment, and interestingly enough, the T regulatory compartment. However, in this case, the CRC tumor is negative on the tumor cells for PD-L1 expression. Now, we've looked for PD-L2 expression by flow. It's been hard to detect, but by the RNA level, we can see that it is expressed. It's not expressed as many cells as PD-L1, but it is expressed by a few T regulatory cells as well as some of the myeloid cells within this tumor. TIGIT, on the other hand, is expressed much more highly and much more broadly than PD-1 is in this specific colorectal cancer sample, and it's expressed pretty much across the entire T cell compartment. Again, it's restricted to that compartment, doesn't seem to be expressed by any of the other immune or tumor cell subsets. In this case, we can define expression of PVR. Again, it's not to nearly the extent of PVR-L2, but it is expressed by a few of the tumor cells as well as some of the myeloid cells. But the main ligand expressed for TIGIT is PVR-L2, again, restricted almost entirely to the myeloid compartment as well as to part of the tumor compartment. So this data set really allows you to look at all of the co-stimulatory, co-inhibitory molecules rather than being restricted to just a few. These technology then can be used to either do barcoded antibodies in CytSeq or back to flow cytometry to have orthogonal assays to really evaluate this at the protein level. Finally, looking at single-cell TCR repertoire data, looking at sorted CD8 T cells from a renal tumor, we do see a high degree of clonality. We've seen this actually in all of the CD8 T cells that we've sorted, such that oftentimes it's 10% or greater of the CD8 T cells have a single clonotype. This is really interesting and really great for those folks who are interested in T cell repertoire analysis of T cell therapies, as it allows for the identification of potentially immune-reactive or tumor-reactive T cells. So throughout this talk, I hope we've been able to discuss how we use Miltenyi platforms to phenotype our donors from our apheresis clinic for downstream isolations and potential use in cell therapy manufacturing. How we use, again, the Miltenyi platform to characterize our DTCs at a high degree for both the use of sample selection as well as for identification of new and novel targets. And then finally, how these DTCs can be used in single-cell RNA-seq and other single-cell technologies to, again, identify novel biomarkers. Thank you.